In this session, we are going to study the chemical structure of DNA, anti-parallel nature of DNA, and we will also discuss about that why purine pairs with pyrimidine, and the double helix model of DNA, and finally the different forms of the DNA. Chemically speaking, DNA consists of three things. It is the sugar, second is nitrogenous bases, and the third is phosphates. So one by one we will discuss about all these three things. So let's first of all talk about sugar. There are two forms of sugar. One is deoxyribose sugar, second is ribose sugar. Deoxyribose sugar occur in case of DNA and ribose sugar occur in case of RNA. So first let's talk about deoxyribose sugar. Now deoxyribose sugar is a 5 carbon structure. Now oxygen bridge present between carbon number 1 and carbon number 4, as we can see in the diagram. This is the carbon number 1, this is the carbon number 4, and oxygen bridge is present over there. The most important thing in case of sugar is the positioning of the carbon number 2. At the carbon number 2, in case of deoxyribose sugar, there is a 1 hydrogen and 1 hydroxyl, whereas in case of ribose there are 2 hydroxyl group. So it is the carbon number 2 which differentiate between deoxyribose sugar and the ribose sugar. Now let's talk about the nitrogenous bases. Now there are two types of nitrogenous bases. One is the purine and second is the pyrimidine. Now purine is a 9-membered double ring structure, and in purine nitrogen is placed at a specific position. These are the carbon number 1, the carbon number 3, the carbon number 7, and the carbon number 9. Similarly, in case of pyrimidine, again it is a 6-membered single ring structure, and here nitrogen is present at carbon number 1 and carbon number 3. Let's talk about the different types of purine. Now there are two different types of purine that exist. One is adenine and second is guanine. Now both adenine and guanine can be differentiated on the basis of the different functional group. For instance, in case of adenine, the amino group is present at carbon number 6. As we can see there, this is the amino group that is present at the carbon number 6. So chemically speaking, adenine can be written as a 6 amino purine. In case of guanine, the amino group is present at the carbon number 2, and at carbon number 6 there is a keto group. Now let's talk about the pyrimidine. There are three different types of pyrimidine. One is cytosine, thymine, and uracil. Now in case of cytosine, the amino group is present at carbon number 4. This is as per the modern nomenclature. In the classical nomenclature, the numbering is done like this: 1, 2, 3, 4, 5, and 6. So as per the classical nomenclature, the carbon number at carbon number 6 amino group is present. Now thymine is very special. Why? Because methyl group is present at carbon number 5. So thymine is a methylated nitrogenous base. And in case of uracil, keto group is present at 2 positions. One is carbon number 2 and another is carbon number 4. Now phosphate unit is present either in the form of phosphate ion or phosphoric acid. It is the phosphate which imparts acidic nature to the DNA. The lone pair of electron that is present in a phosphate is the reason behind the acidic nature of DNA. Now let's talk about that how these three molecules together join and they form a DNA molecule. So we can study all these things in a stepwise manner. So let's discuss the very first step. The first step is the formation of the nucleoside. Now for the formation of the nucleoside, sugar binds with a nitrogenous base and they form a nucleoside. Now as we can see in the diagram, So this is the nitrogenous base, and this nitrogenous base to bind at carbon number 1 of sugar. So the structure formed is called as a nucleoside. In a step 2, we will see the formation of nucleotide. For the formation of a nucleotide, one nucleoside binds with a phosphate group. So nucleoside and phosphate together form nucleotide. Now here also the binding is very specific in the nature, as we can see there that this is the nitrogenous base which were bounded to carbon number 1, but the phosphate group is bounded to the carbon number 5. In step 3, we will see the formation of a dinucleotide. Earlier, we have seen the formation of mononucleotide. So this is the mononucleotide. So the two mononucleotide join together, and the moment mononucleotide join, they form a dinucleotide structure. So both these molecules join together with the help of these phosphodiester bond. Now the phosphodiester bond is formed between carbon number 3 and carbon number 5 of nucleotide. As we can see there that this is the carbon number 3 and this is the carbon number 5. Later on, the polynucleotide is going to be formed. For the formation of polynucleotide, what will happen? That the mononucleotide will keep on adding to the existing dinucleotide, and this addition is going to occur with the help of phosphodiester bond. So in this manner, the chain keep on growing, and as a result, polynucleotide chain is going to be formed. The important thing is that the chain has a two end. One is 5' second is 3'. These 5' and 3' is recognized on the basis of the free hydroxyl group at the carbon. So this is the 5th carbon, so that's why it is called as a 5', and this is the 3rd carbon, so that's why it is called as a 3' end. Finally, the two polynucleotide chain join together, and that joining is going to occur with the help of hydrogen bonding. So the moment they join together, complete DNA molecule is going to be formed. The important thing is that one chain is growing in 5' to 3', then another chain is growing in 3' to 5'. The nature of hydrogen bond is also very specific. Adenine and thymine will form in between 2 hydrogen bonds, and 3 hydrogen bonds will be formed between guanine and cytosine. So this is how a complete DNA molecule is going to be formed with the help of its 3 basic constituents, that is sugar, nitrogenous base and phosphate. Now DNA strand is antiparallel. Why it is antiparallel? Because one chain is going 5' to 3' and another chain is going in 3' to 5'. The chain grows in such a manner because it facilitates the formation of H bonding between the nitrogenous bases. So that is why we call DNA strand is antiparallel. Now, why purine always pair with pyrimidine? The reason is that there is a fixed distance between the two strands of DNA, that is 20 angstrom, and this fixed distance is going to accommodate only a 3-ring structure. Now this 3-ring structure is possible only and only if a purine is going to pair with a pyrimidine. Suppose 2 pyrimidine joins, then the gap between 2 pyrimidine is too big to form a hydrogen bonding. So that's why the 2 pyrimidine cannot present in between the 2 strands. Similarly, if there is going to have a 2 purine, then it going to be too tight that there won't be any space for the formation of hydrogen bonding in between them. So that is why purine is always going to pair with a pyrimidine. Now let's conclude. So DNA structure consists of nitrogenous bases, sugar and phosphate. In DNA molecule, adenine pairs with thymine by 2 hydrogen bond. Guanine pairs with cytosine by 3 hydrogen bond. Adenine pairs with uracil in case of RNA. Adenine and thymine and guanine and cytosine pairing occurs to satisfy the bonding configuration between the nucleotides. Now let's talk about Watson and Crick model. The moment we understood the chemical nature of DNA, now our task is to understand that in 3D sphere how these molecules are arranged, which is often explained through a model. From time to time different model has been given, but the most accepted model was given by Watson and Crick, which is popularly called as a double helix model. This model is based on two most important studies. These are